How does your immune system know when there's an invader? Well, many of our cells throughout the body have the ability to present antigen to lymphocytes. And some of them are actually very good at it, in that they're called antigen-presenting cells or professional antigen-presenting cells. So what these guys do is they're out there looking. They're looking for bad guys. They're looking for exogenous antigen, aka foreign substances. And they're in your connective tissues, they're in your lymphatic tissues, they're out there looking. Some of the most common antigen presenting cells are the macrophages. So let's pretend this cell is a macrophage. It's an antigen presenting cell, or APC. Its job is to go out there, find an antigen, phagocytose that antigen, break down that antigen into pieces to kill it. And that's great. We just killed one bacteria. But the idea is, if there's one, there's more. And this antigen presenting cell doesn't have the ability to phagocytose them all. It needs a lot of help. So it's going to process these pieces and present them on the outside. So we don't need to go crazy here, but you'll notice there's this green thing called an MHC2 molecule. We'll see MHC1 in a minute. MHC2 molecules are proteins that antigen-presenting cells have that can grab a little piece, a little protein, or a little fragment of a protein from that bacterium or virus and present it on the outside, kind of show it. And we're going to see in a few slides, one of your lymphocytes is going to come over here and see that. Say, oh my gosh, look at that. Where'd you find that? And this macrophage says, well, I went and found it, chopped it up, killed it, and this is what it looks like, or at least a little piece of it. This is what it looks like. And then that lymphocyte's going to say, okay, let's amplify. Let's boost the immune response and look for more that have this same little protein or little fragment on them. That's the way we're going to know that that's that bad guy. So this is known as antigen presentation. Notice we've got an APC, a macrophage, using MHC2 to present the antigen. And we're going to see in a little bit that it presents this antigen to the helper T cells. Helper T cells are the ones that come over here and say, wow, look at that. All right, let's do this. We're going to go amplify the immune system and look for more. There's also what's called MHC1 molecules. Very similar in the whole idea, it's antigen presentation. The difference is it's not a macrophage. It's actually all cells of your body can do this, except red blood cells. They're the one exception. They can't do this. But respiratory cells, digestive cells, cells of your body that get infected. All right, so now we have an endogenous antigen. We've got a virus. Let's pretend this is a virus. That virus, let's say it's the flu virus, infected this cell. Uh-oh, that's bad news. At some point, we're going to have to destroy this cell, and we will. But in the meantime, how do we know that this cell has a virus? How does the immune system know that there's a virus inside of this cell replicating, about to destroy this cell and spread to neighboring cells? Well, the only way we know is this cell, in a last-ditch effort to save the rest of its neighboring cells, it's probably going to release interferon, but it's also going to present. But it uses MHC1. It's just a slightly different form of this presenting protein. So it grabs a little piece and eventually presents that antigen on the surface, kind of showing everyone, showing the immune system, oh gosh, look at what happened. This thing is inside of me. You can't see inside of me, but I'm showing you what's inside of me. It's this little virus. So the difference is the type of cell is now any infected cell of your body. The MHC is MHC1. And the big difference is who is it presenting it to? It's going to present to a cytotoxic T cell. So now a cytotoxic T cell comes over and says, oh, geez, I'm sorry to hear this. You have, this purple virus in you, and it looks like that. All right, we're going to go look for more, but before I go look for more, I have to destroy you. Okay, so that's the way that our immune system knows we have to destroy this cell because it's harboring an enemy. It's got a virus inside of it. We can't afford that virus to keep replicating. So we're going to destroy this cell using a cytotoxic T cell. Then that cytotoxic T cell is going to look for more because probably right next door there's another cell presenting the antigen, and that has to be destroyed too. So this is called antigen presentation. We have to keep it kind of straight. Who has MHC1, who has MHC2, and who do those cells present to? Are they going to present to the cytotoxic T cell, or are they going to present to the helper T cell? If you think back to the previous slide, you don't want a macrophage to present to a cytotoxic T cell because then it's going to get destroyed. That macrophage was just doing its job. It's not infected. This guy, that's a different story. This guy is infected. So we want to destroy this infected body cell. Now let's put it all together. So we're going to look at how do we activate a helper T cell? How do we get that helper T cell to start working for us? And this figure shows us how that occurs. We have an APC. This is an antigen-presenting cell, like a macrophage. That antigen-presenting cell phagocytosed some pathogen. All right, could be a virus, could be a bacterium. It phagocytosed it, it processed it into little pieces, and it presented the antigen using MHC2. MHC2, you can see, is this green structure, this green protein. The purple protein is the antigen. That's the piece or fragment of the pathogen. The helper T cell comes over, and there's an interaction that has to occur. The helper T cell uses its T cell receptor, that's the antigen receptor, to bind to the antigen. Notice there's the TCR on the helper binding to the antigen. The second thing it has to do is it has to use its CD4 receptor to bind to MHC2. So think about what this is. T cell receptor to antigen tells the helper that there's an antigen there. CD4 to MHC2 tells the helper that that's one of our guys, that's one of our APCs. So now we got an APC presenting an antigen. Oh my, okay. That means we've got an issue. We've got an antigen, we've got a pathogen. So the helper T cell will then proliferate into many helper T cells. It'll clone itself. And then it'll go boost the immune system by producing chemicals. The most important chemical produced by a helper is called interleukin 2, IL-2. IL-2 amplifies the immune response. It gets us to really induce an immune response so we can rid the body of this pathogen. At the same time, some of those helper T cells are going to differentiate into memory helper T cells, which are cells that can live for a long time, decades, and lie in wait until that pathogen comes back. And then we'll have a lot of helpers waiting and ready. So this is how helper T cells get activated. So now you got all these helper T cells out there. You got the ones producing chemicals that amplify your immune system. You've got those that are helpers. At the same time, you might activate a cytotoxic T cell. So how does a cytotoxic T cell get activated? Well, it's going to respond when there's an infection that gets inside of a cell. And there we have that presentation occurring. The infected cell presents using MHC1 instead of MHC2. It presents the antigen. A cytotoxic T cell is going to come over, use its T cell receptor to recognize the antigen, use a different protein called CD8. CD8 is on cytotoxic T cells, and it's ability to recognize that that's MHC1. So the same idea. You just got presented an antigen. You recognize the fact that that's an antigen, and it's being presented by someone who has MHC1, which is all our body cells. So those two interactions tell the cytotoxic T cell we've got a body cell presenting an antigen. That means that that cell is infected and we need to kill it. It just so happens it also needs to be helped. It needs to be co-stimulated by helper T cells that secrete interleukin 2. So there's that helper helping by releasing interleukin 2. Once all this happens, the cytotoxic T cell proliferates into many cytotoxic T cells. It's going to kill that infected cell, but then it's going to amplify and clone itself and go out and look for more. So those are the active cytotoxic T cells. And some of them are going to differentiate. They're going to differentiate into memory cytotoxic T cells. So now you got all kinds of active T cells out there looking for this pathogen. You've got memory. So if it ever gets in again, maybe years, decades later, you'll have a strong secondary immune response. This is just a couple ways that cytotoxic T cells destroy infected cells. So there's the infected cell. You see that there's a microbe in there. This cytotoxic T cell, when it recognizes all this, it produces some enzymes called granzymes that might destroy the cell, or it makes some other molecules called granulysins and perforins. So the key here is to recognize that granulysins, perforin, and granzymes are all produced by activated cytotoxic T cells to destroy the infected cells. Sometimes it might release microbe, which isn't a great thing. So you'll want to have a phagocyte there catching any microbe that might have been released from that destroyed cell. In the next video, we're going to talk about B cells and how they play a role in your adaptive immune responses.